The microbiome is a collection of microorganisms living in and on the surface of the body. In recent years, the microbiome has gained popularity in several different fields. For instance, in terms of chronic pain conditions, it's typical that patients will have dysbiosis, meaning that the number and types of different bacteria are altered from that of a healthy state. Still largely unclear is if and how this dysbiosis can contribute to the chronic pain that these patients experience. Our lab, amongst many others, use animals to investigate and answer some of these questions. However, to date, there has been no systematic review completed that assesses this preclinical field of microbiome manipulation in measured pain. Therefore, in this paper, we review every existing manuscript that measures pain in response to microbiome manipulation. In total, there were 97 publications that met our inclusion criteria, with 303 experiments across them. The first paper that manipulated the microbiome of rodents and measured pain outcomes was published in 2006, and the field has grown ever since. Largely speaking, rodents were the only species used, with mouse models slightly more popular than rats. We also found that the main microbiome manipulation employed was the administration of probiotics, or good bacteria, so to speak. Antibiotics and fecal microbiota transplants were used at similar rates as well. A handful of experiments also used germ-free mice, which are mice who have no microbiome whatsoever. We tallied the effects of these manipulations in both naive rodents and injury models. These are just two examples of how we reported our findings. On the left is an example of the outcome of antibiotic treatment on naive rodents. Split based on the pain measure used, you can see that when administered to naive rodents, antibiotics most often increased pain or had no impact. Very rarely did they decrease pain. On the right is an example of outcomes in just one group of pain models. Non-GI neuropathic pain models were grouped together, which includes diabetes, traumatic nerve injury, and chemotherapy. The specific manipulations are reported, and effects on pain based on the measure used are also shown. Overall, we found that manipulations of the microbiome affect both cutaneous and visceral sensitivity. We also found that there are differences based on whether or not these manipulations were performed in naive or injured rodents. In naive rodents, it appears that microbes contribute to a body-wide antinociceptive tone. However, in injured rodents, it appears that more often than not, when treated with antibiotics or probiotics, their pain is actually decreased. Lastly, the field is moving into more mechanistic approaches by performing fecal microbiome transplants, either from rodents who are in pain to rodents who are not in pain, in addition from human chronic pain populations into rodents who are not experiencing pain.
In completing this review, we identified several gaps in the literature that are immediate and exciting research opportunities for those working on the microbiome and pain. First, and perhaps most shocking, there are no studies that have measured cutaneous touch, heat, cold, or chemical pain sensitivity in uninjured, germ-free rodents. Second, even though the vast majority of experiments in this analysis measured pain by poking, cooling, heating the surface of the body, we have very little understanding of how skin microbes actually contribute to these sensations. And lastly, it is unclear if the effects of individual antibiotic drugs or probiotic bacteria work in similar or diverse ways to change pain sensitivity. Our lab and many others are interested in answering these types of questions. If you want to learn more, check out our website or reach out to me and we can find a time to chat.